Okay, we better make a start. Hello, good afternoon, everyone. My name is Yusuf. I'm a consultant rheumatologist and also the BRC Academic Capacity Development Emerging Lead. I'm here to moderate the session on behalf of my colleague, Dr. Carol Burnett, and also Professor Anne Henry, both the ACD leads, and also on behalf of the ACD committee. So today we are proud to host an important topic. So this is the series number 7 of this calendar year. So the topic today is clinical trials, so baskets, umbrellas, platforms, and all things adaptive. So just before I proceed to introduce the speaker today, I just want to share you our, spare a second on the screen. Yeah, so I just want to share you our QR code for today. So we would like to hear from you, what things go well and what things could be improved. So there's a QR code there if you want to snap it. But also, I'm going to share it at the end of the session. And after the lecture finish, we also will email you separately as well as a reminder, with a link in case if you can't bring your phone to you today. Yeah, so and we also like to know, what are the topics that you want us to include for future references. Okay, so a few house rules today. So automatically, everyone have been put on silence. So can you please not unmute yourself during the presentation because we don't want any echoing to ensure smoothness of the program. And also, if you do have any questions, please put on the chat. At the end of the session, I will moderate this and we'll read, the questions that you pose. And if anything that we can't be able to read your questions before the time, you can always, discuss that with, Deb at the later time. So without further delays, I would like to invite, and we are so honored to have Professor Deborah Stocken. She's a Director of Surgical Intervention, Diagnostic and Devices Division at Leeds Institute of Clinical Trial Research to deliver her talk. Thank you.
Thanks, Yos. So first things, I'm just going to share my screen and get that working. Can you confirm if you can see that?
Yes.
Yeah, it's working. Okay, brilliant. So because I've shared my screen now, I can't see anybody, but I think yes, we'll have questions at the end, and I'm really happy for anyone to contact me afterwards as well. So first of all, just thank you to yours, Carol and Anne, and the Leeds BRC for inviting me to talk to you today. So for those of you who don't know me, I'm an NIHR Research Professor in Statistics here at Leeds, and I'm also Deputy Director for Research at the Leeds Institute for Clinical Trials Research. And I've been invited to talk to you today to share some concepts and ideas about adaptation in clinical trial design, how it all kind of started, how it began, and hopefully with plenty of references so you can go off and do your own reading. And as I've said, yeah, please ask questions at the end. And if anyone wants any further information or a chat afterwards, then I'm very happy to do so. Okay. So you can see from the graph here, this is a paper that was published in 2018. The timeline on this graph goes from 1978 up to 2014. And so you can see that adaptive methods have existed for essentially decades. This 2018 article demonstrated the growth of adaptive trials worldwide. As you can see, they emerged in the 1970s, and they were particularly important at that time in industry-funded research, and predominantly with investigators from North America and Europe. But you can see later on up in the middle of the graph here, I don't know whether you can see my pointer actually, but with rapid growth from the 2000s onwards. But let's just take a pause and think initially, what is adaptation? So there's a really good paper that came out of the MRC network of hubs for trials methodology, the Adaptive Design Working Group, that described adaptive designs in this 2018 paper.
That's great, isn't it?
Oh, Thank you. I think we've just had a baby join. And you can see from this 2018 paper that adaptive trials are described as flexible designs compared to the top row of this table here, which is the traditional trials with fixed design. So a fixed design would design a study, conduct a study, analyze study. The more flexible design, the idea is that you design the study, and during that conduct phase, you might interimly review or adapt the trial with a pre-planned adaptation and consequence. So the flexibility comes from the planned review during the conduct phase, and there is pre-planned prospective consequences of that review. And in essence, at any predefined stage, the trial data may be analysed, and the next stages of the trial might be redesigned, taking into account all of the available data at that time. The first formal definition came from a white paper from the FDA in America back in 2006. They called for more innovative designs to speed up particularly drug discovery and the drug assessment pathway to try and get the right drugs to patients more quickly. We all know how long trials can take. So they wanted decision making to consider the use of prior experience, and they also wanted the accumulating data within those active trials to be accounted for and assessed. So they specified the need for robust decision making based on trials being adaptive by design, not, as it states here, a remedy for poor planning or any post hoc adaptation. And I think this is probably the most crucial aspect to the definition of adaptive trials, that it is adaptive prospectively by design. So to expand on this, their definition stated that the accumulating trial data could be considered where methods did not undermine the validity and the integrity of the trial. So by this, they mean in terms of validity, controlling for statistical errors and statistical bias, and for integrity, considering the clinical utility of the trial results and being able to convince the broader scientific community of those trial results post-adaptation. So back in 2007, this is one of the first books that was published by Chow and Chang, outlining the different types of prospective adaptation. I'm going to touch on some of these today, but obviously not all of them in the interests of time. But importantly, you can see how adaptation is relevant across all phases of clinical trials. So by that, I mean from phase 1 dose escalation trials through to adaptive randomization, sample size re-estimation that we often use today in our phase 3 studies. So right across the phases of trials. And if we go back to the graph that I showed you earlier, but if I now show you the different types of designs on the bottom, I know it's quite small, the text, but you can see how some of these different adaptations have increased over time. So for example, you can see how the seamless phase 2, phase 3 designs, which are in light blue and start in the 1970s, you can see how they've escalated from the late 2000s. They start escalating in number, and how the phase 1 adaptive dose finding and the drop the loser designs start to appear in the middle of this graph in the 2000s, and how something we call group sequential designs take off after about 2010, and I'll touch on some of these today. So we can have adaptation through the phases of trials from phase 1 dose finding to phase 2 efficacy and up to phase 3 comparative studies. Starting at phase 1, the most common adaptive method at phase 1 is the continual reassessment method. And this is where the initial tolerated dose is estimated and the dose response model is updated with each patient cohort following treatment. So the cycle is repeated until either the statistical model is converged or the maximum number of the preset maximum number of patients in the trial has been reached. This is sort of what it looks like in practice. You can see the graph on the right. The CRM methodology was first published in 1990, but it took another 15 years for it to make it onto that previous graph I showed you. This graph on the right shows how the model is updated from, you can see the black line, which is the prior curve with a probability of dose-limiting toxicity set at 20%, and the anticipated dose level at the start of the trial, the prior expectation was that dose level 1 would give that 20% dose-limiting toxicity. But you can see how with each cohort of patients, this curve moves, and you can see at the top of the graph how those lines start to converge, and they converge by cohort 9. And if I tell you that each cohort has 3 patients in them, that's 27 patients in total for that statistical model to converge. And all patients that are recruited within each cohort are included in each analysis at each time point. And the main advantage over other phase 1 methods is that you can actually estimate the probability of a dose-limiting toxicity with a confidence. And I think one of the most important advantages is that there are more patients treated at the optimal dose as that statistical model starts to converge. However, it does require a lot of statistical simulation. The CRM method here at phase 1 does start to introduce
the aims of getting the carbon footprint for the different pathways.
Okay. Sorry, could you just mute your microphone, please? Thank you. Interesting conversation about carbon footprints. So we start to see here the CRM method in the phase 1 dose escalation phase of trial start to introduce the idea of multi-stage designs where patients are starting to be analysed after each stage of recruitment. So there's many other adaptations that are more relevant to phase 2 and phase 3 trials, and I've selected these 4 areas to share in a little bit more detail to hopefully try and describe the evolution of the multi-arm, multi-stage, sustainable basket, umbrella and platform trials that we'll get onto later. So this is the usual single arm, single stage early phase 2 trial that you may be used to, estimating response to a new treatment where a priori decisions are made specifically around to specify the thresholds which require further justification of that drug in maybe a later phase 2b randomized trial or a late phase 3 study. Patients are recruited in a single arm and are all given the new treatment, and the thresholds that inform whether that's interesting enough to take forward are informed by historical data or current knowledge of a current standard of care. Multi-stage adaptation can be introduced into these single-arm phase 2A trials by using methods that were first published in the 1980s and 1990s. So Simon suggested a single-arm, 2-stage design based on a single outcome of response. This was adapted in the 1990s for dual outcome of response and tolerability, which you can see in the table on the right-hand side. And both of these methods are examples of where early stopping for futility at the end of the first stage of recruitment of patients is built into the design, and else continuation of recruitment continues into stage 2. A further iteration can adapt the number of patients at stage 2 based on the response that you see at stage 1, and this starts to introduce the idea of an adaptive sample size adjustment. So this is an example of a randomized single stage early phase 2B trial, which you might be used to, where again, we're looking at potentially the response of a new drug to see whether that it justifies further investigation in a phase 3 trial. But in this randomized phase 2B, the anticipated benchmark of the current standard of care or the control arm is actually evidenced within the trial itself using a prospective control. And the reason for this is to reduce bias. So sometimes the benchmark standard of care is evidenced with less patients, and we might use an unequal allocation. But you'll notice that this design does look a little bit similar to a late phase randomized phase 3 trial. And one of the risks of this design is the direct comparison of the arms. So just a quick reminder that statistical designs for early phase randomized phase 2 trials don't test the equality of interventions, but rather aim to select promising interventions to take forward for late phase testing. So this kind of standard process of a phase 2 trial being conducted independently of potentially a later phase 3 trial doesn't always feel very efficient. So a standalone phase 2 trial is justified where maybe there's still some learning required, such as the actual patient cohort to be recruited may not be confirmed, or there may be some intervention development or outcome confirmation with changes to these may be required at the end of phase 2. And I think this really does justify a standalone phase 2 trial. But if not, it could be appropriate to seamlessly transition through from randomized phase 2 into a randomized phase 3. And the decision point at the end of the early phase 2 component may be based on response data, as we saw earlier, and that might be a surrogate of a longer-term clinical outcome. Else the decision could be based on a planned interim analysis or analyses of the primary outcome of your phase 3 component of the study. And this might be using something called a group sequential statistical design, which is an adaptive multi-stage method and allows you to perform interim analysis or analyses and make decisions about whether to continue or not. So this adaptive multi-stage approach can be a very efficient approach if we want to formally terminate interventions which cross a formal boundary for futility or safety. We can introduce adaptive sample size adjustment to ensure the precision of an estimate of a final treatment effect. Although multi-stage designs do require additional statistical and operational planning, they do efficiently account for the interim looks of the data, and they efficiently combine data across the stages. And the adaptive sample size adjustment has gained traction recently, where planned formal interim analyses can inform the total sample size that's required to achieve a powered comparison. I'd just like to give a bit of a warning here that sample sizes in sample size adjustment can go up as well as down, and often this isn't recognised, and that can be an issue in relation to funding. Also, a little bit of caution that sample size adjustment is initially based on that smaller snapshot of your accumulating data, and this is inherently associated with higher statistical errors. But when designed correctly, it will result in an adequately powered trial that recruits the smallest number of patients that's needed to demonstrate a real benefit if it truly exists. And in my experience, response adaptive randomization is less well utilised, and this is often because of the operational aspects that are associated with it in terms of implementation. So interim data can inform whether an arm is or appears to be doing better, and the allocation of future treatments can then be biased in favour of that arm. It does rely on adequate interim data informing that differential effect, and it does mean that you need to have the ability to amend the protocol with sufficient time and sufficient patients remaining before the end of your trial. And sometimes, in my experience, that's been a bit of a barrier. If we take all of these adaptations together, in this example, again, the text is a little bit small, but down the right-hand side here, you can see this is the conventional trial at the top with the sample size reassessment underneath, and you can see how the increased sample size had taken effect by the final analysis stage at the end here. The third line here is adaptive randomization. And the bottom one is about enrichment design, which I haven't been into today, just because of time. But if we applied all these adaptations, essentially, we would end up with what we would describe as a multi-stage, sample size adapted, response adapted, randomized enrichment trial, which is a bit of a mouthful, but is obviously very adaptive. And I think just a word of caution at this point to say that although adaptive designs are around and may be applicable, it does not ensure that you can deliver the trial accordingly. And I think it's really important that any adaptation, that you give confidence to funders that you're able to actually adapt and implement the adaptation successfully. So the different adaptive studies that I've mentioned so far have all been related to a single intervention, and the next step in the process of efficiency is to consider where multiple research questions and interventions can be answered within the same trial. The key efficiency in this design of a multi-arm phase 2 or phase 3 trial is the multiple use of the control arm for multiple research questions. Usually the design will compare each of the interventions you can see here in green, U1, 2 and 3, with the control or the standard of care in blue. Less often are interventions compared against each other, and this is since the differences between the intervention arms are often smaller and more difficult to detect. So one of the first publications describing multi-arm trials was back in 2016. This was from Saville and Berry, where the notation of umbrellas and platforms were used. So the umbrella here refers to a single trial of a single intervention in multiple patient subpopulations under what they call an umbrella protocol. A platform trial was described here as a single trial of multiple interventions in a single patient population. The combination of the two, so a single trial of multiple interventions in multiple patient populations, was described in this paper as a platform umbrella trial. And the term basket appeared a little bit later. This systematic review that was published in 2019 showed the first basket trial to have been conducted in 2001, the first platform trial, according to their definition, in 2005. And you can see from the graph the escalation up to 2019 of approximately 80 trials running under what we call a common feature of a single master protocol. And interestingly, approximately 50% of these trials in this systematic review were of single interventions. They weren't multi-arm. So this updated paper from 2019 demonstrates the expansion of platforms from multi-arm into multi-arm, multi-stage definitions. So in this platform, in this paper, a platform trial is described as having planned interim analyses using either the grouped sequential designs or other designs being utilised, resulting in that multi-arm, multi-stage design that we now commonly know as a MAMS trial. And note in this updated publication, the multi-arm trial in a single patient population is now an umbrella, not the platform from previously. The trial of a single intervention in multiple subpopulations is now here a basket, not previously the umbrella. And I think this just highlights that people are often using different terminology to describe different platforms. And it's useful that you're aware of the fact that people are using maybe different terminologies. So the systematic review highlighted the MRC Stampede trial as the first platform. That was the 2005 study. This trial opened to recruitment in 2005 and formally closed following analysis in 2014, as you can see here. It was a 6-arm, 5-stage MAMS design, multi-arm, multi-stage design with planned interim analyses for futility. Patients were assessed for a total of 15 years in the trial, and the authors estimated it would have taken over 40 years had the trial been sequential, not simultaneous. And the Stampede trial also at this point introduces the final concept I want to talk to you about, which is sustainability. So sustainability is shown here and is where new interventions are planned for inclusion in a MAMS design prospectively, making the trial sustainable for the longer term. So in this example, one of the interventions, this intervention C, has shown to benefit to the point of changing standard of care. So at this point, at the third interim analysis, changes to become the new standard of care. And at the first interim analysis and second interim analysis, 2 new arms are introduced as pre-planned, at the pre-planned interim analysis time points. So this brings in the idea of sustainability and long-term longevity. So sustainability is a key element of some of the NIHR funded platforms. This is an example of the current call, which is open, the Australia UK platform call, and it states, and I'm just going to quote from the screen, where there is a pipeline of technologies which justifies a platform approach. So this a platform here isn't just a multi-arm, multi-stage design, it's a multi-arm, multi-stage with some sustainability. The detail for this call is here. The multi-arm trial of multiple interventions under a master protocol, terminating arms with a lack of efficacy at interim analysis. So again, a MAMS approach, but a requirement as part of the brief for a sustainable pipeline of interventions, which can be added as identified. So I think the efficiencies of platform trials, I hope, from what I've talked to you about, are quite clear. There's an obvious benefit of time as we're simultaneously exploring multiple interventions in the same study. There's an obvious benefit in a reduction of number of patients due to that shared control arm, and also the potential for arms to drop early if they're not showing benefit. The multi-stage aspect allows us to pick the winners and also, more importantly, drop the losers based on accumulating data throughout the trial. The obvious advantage of being able to incorporate new interventions as they become available, and opportunities methodologically to look at non-historical controls and utilise that information in your trial design as well. I hope I've shown you that the adaptation is applicable across all phases of trial, but really important to know that there are different statistical methods for early phase trials, and this should be reflected in the design and the sample size if you're designing an early phase platform as opposed to a late phase platform. And also the opportunity to seamlessly move from early phase to late phase, but sometimes this is dependent on whether the funder allows it. I know the Australia call, it has to be very clearly either a phase 2 platform or a phase 3 platform, and they won't fund seamless pull through from phase 2 to phase 3 in that call. So the benefits can be many. Conduct, it can be easier with a master protocol to cover all of the different interventions in your study. Once you've actually developed it and written it, there's an obvious benefit of having that shared infrastructure in relation to the randomization programs, the database, the data collection, and an obvious benefit of having the same staff involved in that platform. So you've got that shared knowledge and shared learning. I think that the platform, the PPI work that we've done with some of our platforms show that the patients find these quite attractive, due predominantly to an increased probability of being able to be allocated to one of the active interventions. I think one of the main drivers for patient benefit is that we get faster results and are able to change practice more quickly through these platform approaches. There are multiple development opportunities. Platforms should be designed as inclusive in relation to the clinical teams. So the funders often require different CIs for the different research questions, and there's an obvious role for the early career researchers to be pulled through and supporting the platforms. And there's obvious development opportunities in relation to the methodology of how you conduct these trials and the statistical designs of the trial. And there are benefits of cost by running multiple trials within one study. But there are challenges, as we know. So a multi-arm, multi-stage sustainable platform will have many challenges, not only due to the complex design of running multiple trials with multiple interim analyses, but also the operational conduct and how we share the acknowledgement. So one of the challenges can be potentially bringing industry partners together under one shared master protocol. There is a real upfront statistical simulation. It's very consuming to design these studies when you've got multiple interventions, and they often incorporate multiple different designs and research questions within those specific randomizations. There's extensive complex statistical simulation for the operational design of the study. Again, I'm going to come back to deliverability and feasibility. So how much adaptation is actually deliverable? At the end of the day, your study does need to be successful in terms of recruitment. Adaptive protocols need approvals to be changed when new arms are added or standards of care change. And there is intensive and ongoing data management and ongoing statistical analyses involved in those interim analyses, which require modified statistical analysis plans. When do you publish? Because you've got lots of interim analyses and an ongoing trial, so that can be a challenge. And also we find that the publications are open to additional scrutiny from the journals and from the reviewers. And how do you actually author some of these outputs, given the huge team that you assemble to be able to design and deliver a platform? And although their cost, they can be cost efficient, the cost can actually be equally prohibitive. I'll come back to that in a minute. So in terms of the challenges around conduct, there has been a piece of work that's been conducted by the UK CRC registered network of trials regarding the operational aspect of running specifically late phase platform protocols. This practical guidance was published in 2022. I'm proud to say Leeds Trials Unit was one of the 6 CTUs with platform experience to be able to contribute our lessons learned to this publication. The guidance includes 17 recommendations and it covers the entire life cycle of the platform. And so I'd recommend that's a good read. I can't go through all of that today, obviously. But coming back to cost, the group, the Trials Unit group, also included a snapshot of the elevated costs of running a multi-arm, multi-stage sustainable platform. And you can see here from the table in their paper that at least twice, if not 3 times, the number of CTU staff are needed to deliver these platforms. And this excludes the consideration of costs of multiple CIs for the different research questions, as is often recommended by some of the funders, as well as any blinding costs for blinding drugs or any excess treatment costs. So although they are cost efficient, they are more expensive, as you would expect. The economic costs have been formally assessed, and again, a paper in 2022 reported, and I'm just going to quote, that despite having larger initial setup requirements, a single platform trial can drastically reduce costs and efforts, highlighting the need for dedicated source funding, and hence some of the commission calls that we're starting to see coming out today. So data from the end of last year showed that NIHR had 22 active or contracted platform trials in place. Half of these were full platform awards. 10 were 12-month acceleration awards, which are awards for working up a platform. Of the 11 full platform awards, 2 were COVID platforms and 5 were funded by the HTA's, the Health Technology Appraisal Scheme, indicating that these are late phase platforms. And the initial duration, as you can see here, and this is without any extensions or variations to contract for longer term sustainability of a platform, the initial duration was for 5 years on average, with an average cost of £6 million. But you can see the range here was up to a whopping 21 million for one study. So I'm just going to pull things together a little bit to I'm going to use this EU Pearl summary. This is the EU Pearl initiative, which stands for the European Union Patient-Centric Clinical Trial Platform. So the summary from them They looked to discuss the outlook and challenges for platform trials in Europe. It was in collaboration with patient representatives and the EMA and the FDA. And they summarised that the 5 ingredients critical for attaining the full potential of platform trials for patients is, first of all, having a pipeline of interventions in the development, having a network of sites to be able to deliver the platform successfully, having consensus on the endpoints up front, strong and involved patient advocacy, and upfront identified recruitment challenges. And I'm just going to summarise by coming right back to the start about any key requirements for any good clinical trial. So any good clinical trial, whether it be a platform trial or another adaptive trial or a simple 2-arm study, the research question should always be important to the clinical community and the patients. We should always have designs that are efficient. We should have research that is deliverable, and we should have research that results in quality and unbiased evidence for patient benefit. And I quite like this quote from Winston Churchill. And you could put research in here, no research idea is so outlandish that it should not be considered with a searching, but at the same time, a steady eye. And I'll end there, and I'll thank you very much for inviting me to talk today. Thank you very much.
Okay, thank you. Fantastic, Professor Stocken, for the talk. So I'm just letting the people, attendees, so if you can post any question in the chat, then we can read the questions, or potentially you can raise your hands as well, that I may allow as well. Just to kick off the Q&A session, since you've also been on the other side, as the funding panel member, I'm just wondering how do you can you leverage in terms of trying to convince the funding committee about the sample size if you're using this adaptive, in case you may need to increase your size after the interim analysis. So how do you leverage that?
Okay, so from a funding point of view, we do see designs coming through with a sample size re-estimation built into them. And often, as I said in my talk, this is around estimating sizes down. And sometimes that sample size re-estimation can elevate a sample size. So we always ask the applicants for the worst case scenario. So what you'll know about your intervention, what you know about the important differences. If you see, one scenario where maybe the arms are closer together than you anticipate, but it's still clinically relevant versus, a larger difference, what impact that would have through kind of statistical simulation at your interim analysis point, and what the impact would then be on your second stage of recruitment or further stages of recruitment. And really what we want to know as a funder at a committee is what the worst case scenario is. You don't want to know the best case, you want to know the worst case. And if that's a real worst case scenario, then you need to share that, because the worst thing is that you come back 2 years after funding and try and double your sample size, and the funding committee may really push back on that, and then that's it brings into account, it brings into consideration ethics, the ethics of conducting that first stage of your trial if you then can't complete the trial. So yeah, we'd always urge applicants to give us worst case scenarios, and also those kind of stopping rules where they would stop because a trial becomes sort of unfeasible to deliver, essentially.
Thank you, Deb. So I think we're still waiting for more questions from the chat. I'll have a different question. There's more question here. So Rosie Beckett asked questions. So what is the benefit of a platform study which utilizes entirely different protocols alongside a main protocol rather than a single protocol that include multiple arms?
Just say that again, sorry. What's the advantage of having?
So she's asking the benefits of a platform study which utilizes entirely different protocols alongside a main protocol rather than a single protocol that included multiple arms.
Okay, so I think part of some of the NIHR definitions of a platform is that you have this master protocol. So I haven't been involved in a platform study that hasn't sort of had that master overarching protocol. And I think part of the reason for having that is around the shared infrastructure that you have. You know, protocol is the Bible of how you conduct your trial. It's very operational, and part of that is the operational aspect of data collection and planning, as well as the kind of academic kind of sample size and design, the methodological design. So I think the advantage for having the master protocol is really clear. I think it would be more confusing to have multiple protocols. I can see having multiple protocols if you were running, you know, the same protocol in a different country, for example. So you might have a trial that's running in the UK and you want to, you know, have the same trial running in, I don't know, Australia, and then you're going to plan to combine the data. But that's a different type of trial, because it wouldn't include your kind of planned interim analyses. And I don't know how you'd conduct the planned interim analyses across countries. So I don't know whether I'm answering the question very well at all, because I don't really know how you would do it without the master protocol, is what I'm saying.
I probably will let Rosie to maybe clarify your question, Rosie.
Hi, yes, sorry. It's a little bit of a confusing one, and it may come back to the fact that people are using a platform in different ways to define study design. So from a hosting perspective, we see platform studies where it's, as you've described, it's one protocol, and within that one protocol, they've made different arms and different processes, and then they adapt throughout and drop arms and add arms as needed. And then we see scenarios where, again, it's called a platform study, and it's one main protocol, and within that, they then add additional protocols, almost like an appendix. So you get one study that's a title like, I don't know, the chocolate study, for example, and then you get individual protocols additional to the main overarching protocol that define a particular way of delivering that appendix study, if that makes sense. So from a hosting perspective, it adds a lot of complexity, because typically you get one HRA approval, but you've got multiple studies under that. You have multiple protocols that refer to a main protocol. And then from just a purely administrative perspective, you've got to log it in a different way from other typically normalized research studies. So it was more of from a design perspective, because I've not been involved in the design of such protocol. Is there a pro or the benefits of that type of study that you're aware of versus just using one single protocol and having multiple arms outside the obvious, well, they can adapt a little bit more with an individual protocol?
Thanks, Rosie. That's much clearer. I think there is an advantage if I can imagine if you have subpopulations. So if you set up a big, you know, platform and you've got a designated sort of patient group. If you think about those subpopulations, if there are some interventions that are only relevant to some patients in that study, I could see the advantage of having, you know, patients randomized into that arm, and then there may be an additional protocol around, you know, the deliverability of some aspect, if that isn't then relevant to all of the other patients in that trial and in that platform. So I can see then that there would be a real advantage where you've got subpopulations, because otherwise you're adapting a protocol for, you know, every centre who's taking part will have to put through a protocol amendment. If you've got a subpopulation, maybe it's a mechanistic population, it might be an EMI platform, it might be an early phase platform, and there's a mechanistic component to it, and maybe it's only 2 or 3 sites are taking part in that because of the kind of exploratory nature of it potentially, then I could see the advantage of having, you know, multiple dependencies of your protocol in that situation. Does that help?
Hi, yes, I understand what you mean. I suppose there's a little bit of crossover between sub-studies and platform studies, because you could very well have that same design that you've outlined with sub-studies. So within a protocol, you can have a certain population that you want to run a sub-study on, but these platform studies that we're seeing more and more of they don't come under classification of sub-study. And again, it may just be the terminology that's used around it. But I just wondered on your thoughts on it.
Yeah, and it might be worthwhile going back to the trial and having a look at whether it really is the kind of platform. You know, the definition now of a platform is really about multi-arm, multi-stage with this potential plus or minus sustainability. If it's not really multi-arm, multi-stage, if it's more multi-arm, it might just be terminology about it being called a platform because it's trendy to have a platform at the minute, but maybe it doesn't fit the true definition.
Possibly. I get there's a lot of complexities in research, isn't there? But yes, thank you very much.
Yeah, no problem. Thanks, Rosie.
Okay, so we've got quite a long question. Actually, maybe probably just better for me just to ask John Turgos if you could just simplify your question to Deb. That would be great.
Hi, Deborah. Thanks for the talk. Yeah, obviously noting and appreciating you're a statistician, but I work in data management, and I'm just asking for some reflections and thoughts if you feel able to provide them on some of the data management challenges with these adaptive designs. And there's there could be a whole range, but perhaps one to think about would be whether to have a single study database that you amend at each time point, or whether you would advocate essentially taking a copy of that database and making the amendment so each time point effectively has its own database as an example, but other reflections welcomed as well. Thank you.
Yeah, no, I mean, it's massively intensive, the data management, because of the planned interim analyses happening, often quite frequently, and they're formal analyses. They're not kind of safety analyses that are going through a data monitoring committee and we're checking safety. This is formal decision making about whether to drop an arm or not. So the data has to be really clean and robust at each of those time points, and that makes the data management an ongoing task, and so they are very, very intensive. In fact, one of the tables, I think it was the data management arm, where I think it was up to 6 times as many people were needed. I'm sure it was the data management, I'd have to check, I think so it's huge, and I think it needs to be adequately costed, because you can't do the analysis and the decisions that are made at those points are only as good as the data that you have at those points. So if you haven't invested properly at that time, then you're going to make potentially the wrong decisions. What was the second part of your question, John? Sorry.
Yeah, it was just around a more practical point around whether you would have a single database, study database, and you would amend it for each new time point, or whether, in your experience in the CTU in Leeds, that you would create essentially a new database for each time point, almost a copy with the adaptions in there.
I think the only. So not for a time point, because essentially you're collecting the same data, on the advantage and the efficiency comes from having a single database that you've pre-planned. One of the Pearl initiatives was around agreeing the endpoints and making sure you've got agreement there. What you don't want to be doing is changing, endpoints and definitions as you're going through the trial, unless you actually have to, and that could be part of your planned adaptation if you need to. But otherwise, you should be collecting the same information across the same arms and the same follow-up, and that brings the efficiency of having a single database. I think where it can become more complex is if you bring in a new agent or a new arm, part way through your study. One, you've got to adapt your randomization, but you can, that will be adaptable. But if there's a safety profile or something that's related to that individual drug that hasn't been applicable before and doesn't appear in your database, then you're going to start then needing to adapt your database and take it to the next version on with additional data collection that you won't have on the patients up to that point. You won't have that information on, so you end up with a version 1 without it and version 2 with it, and I think that's the way that you have to move forward. But I mean, the efficiency comes in having the single database where you can have it. Back to the last question from Rosie, if it's an early phase platform, you will have potentially some mechanistic data, you might have exploratory analyses because of the early phase nature of the trial, and it may be that you have, you know, spin-off databases that are relevant to certain sites or certain subpopulations that aren't then open, you know, for everybody in the full trial. So I can see that happening, but that will be because of the, you know, the early phase nature. So I think it all boils down to the interventions.
Brilliant. I've got another question, Deb, because we still have time. Pertaining the basket trials. So, for example, in rheumatology, we have in a condition called autoimmune connective tissue disease. So basically, we have got various diseases that stems from one positive test called ANA test. So basically, in some diseases, like, you know, they have more advanced therapy, and some, you know, diseases, unfortunately, they didn't have licensed therapy. So if you want to do like a basket trial of like a drug with multiple diseases, how do you take into account in terms of essential import key heterogeneity? You know, for example, like ancestry and things. How do you take into account that?
So I think I'd want to know, when I was designing the trial, you've got various subpopulations. You've got one treatment and various subpopulations. Is that what you're saying? Yeah. So the question I'd want to know as a researcher is, are you planning to, you know, what's the distribution of those subpopulations in terms of, you know, the proportion of patients that are going to come into your trial? If one of those subpopulations accounts for, you know, 5% of all patients, then you've got quite a small subgroup there. And then the next question is, how are you going to analyze your data? Are you wanting to show that a new treatment works generally in this group of patients, but you don't know whether there could be some differential effect? So the main decision about moving forward would be, you know, an aggregated treatment effect, and you take the trial forward into phase 3 or wherever. Or are you looking for to actually identify where those differential effects are? And then that's slightly different because you're looking at the treatment effect within a subgroup. So I'd want to know, are you looking at treatments in a subgroup? Are you looking to look at the aggregated effect, but adjusting by the knowledge that you have different patient populations, and they're slightly different in terms of the question, and then the design will be slightly different depending on what the question is.
Yeah, brilliant. Yeah, that's answered the question. Yeah,
that might need a further conversation offline.
Of course. Yes. Yeah. So while we're at that, I think probably the last one. So in terms of trying to get help and support, would you like to, you know, provide some insights? You know, where, you know, all people who want to, you know, get involved with this adaptive trial, who should we contact, and etc.
Yeah. So the NIHR Research Support Service is always, you know, it's a national support service for a first point of contact for anybody in the country, to be able to, you know, speak to a statistical advisor, a trials unit advisor, etc., etc. So wherever you are in the country, you can access that. If you're already linked into a trials unit, we work very closely with, you know, some key investigators in Leeds and elsewhere nationally. But obviously the trials units directly, if you have that contact, can help. I think it's useful to maybe speak to a trials unit who've got some experience in delivering a trial. You can have the best idea in the world, but delivering it, you know, it has to be deliverable. And I think the operational aspects of platforms are complex, and that's often where some of the expense comes in as well. So yes, the design is complex, but they have to be deliverable. You can have, you know, bringing the funding in is one thing. Actually keeping the funding and recruiting the patients is far harder. And so I think really talking through with a trials unit, with that multidisciplinary team of, you know, data managers, trial managers, people who are going to be directly engaging with site, and the other clinicians who will form part of that network for your platform. You won't have, you know, a single CI. You might have multiple investigators who are leading different research questions in your platform. And I think making sure you've got the network nationally set up for recruitment, and the trials unit, you know, the trial management aspects of it set up for deliverability, is really, really important. So, you know, a first contact and a conversation with the trials unit would be good.
Yeah. So yeah, thank you so much, Deb, for your beautiful talks, and we all really enlightened your presentation today. I'd just like to share the screen again. So for whoever's got a mobile phone, so basically this is our QR code for the feedback survey. So I hope you can provide some feedback to us, and what are the topics that you're wanting for the next time. Once you filled in, it's only take about 2 minutes. You will also get automated certificates, which you can put, for your continuing professional development as well. And just letting you know that the video that will be recordings will be available. So hopefully about 2 weeks from the event today. And just lastly, before we go, just letting you know that we have our last session of this calendar year, because we're going to have a break for summertime, which is going to be in 2 weeks' time on Tuesday, 17 June. So the topic will be conflict and managing difficult conversation. So that will be an interesting one as well. So if you can sign up for that, that will be great. And so thank you, Deb, again for your talk, and thank you everyone for attending today. We got really good turnout and good conversation today, and hope we will continue the conversation about adaptive clinical trials. Have a nice day, everyone.
Thanks for inviting me, Yos. Take care. See you soon.
Bye-bye.
Bye.